Welcome back. Here we are with a session on biochemistry where we are going to discuss on the biochemistry of transfer RNA or the tRNA. Transfer RNA, it is otherwise referred as soluble RNA or the adapter RNA, and it is involved with the transfer of amino acids from the amino acid pool in the cytoplasm to the protein synthesizing machinery which is present on the ribosome. So it serves as an intermediate molecule which can transfer or which can convey the what you call the amino acid from the cytoplasm to the uh, ribosome. Okay, it can be considered as an adapter molecule that can accept the specific or the particular amino acid complementary to the codon present on the mRNA. Now, they make up almost about 5% of the RNA in the cytoplasm, 10 to 20% or 15% of the total cellular RNAs, it is the tRNA. The main function, as already mentioned, they function as the adapters for the translation of the information in the sequence of nucleotides of the mRNA into specific amino acids. What does it refer to? Actually, the mRNA, it is produced in the nucleus, as in the case of specifically in the case of eukaryotes, it is produced in the nucleus, and this mRNA, it is formed of the nucleotide bases. It contains the nucleotide base sequence complementary to the base sequence present on the DNA. Okay, so the information on the DNA, it is being copied onto the mRNA, and this information, it is carried in the form of mRNA to the cytoplasm where the proteins are being synthesized. Okay, now, so when the mRNA passes into the cytoplasm, it contains the information, but there should be some structure or some machinery that can read the messages on the what you call the mRNA, right? And based upon that what you call decoding, there should be some machinery which can take the amino acids complementary to the or corresponding to the information present on the mRNA, and that is done, the decoding of the information or reading of the information on the mRNA plus the what you call carrying the amino acids or bringing the amino acids corresponding to these messages on the mRNA. It is done by two molecules. One is the ribosomal RNA, ribosomal protein complexes, the nucleoprotein complexes, that is the ribosome, what we call. Another is the transfer RNA. Okay, so these two molecules together make up the protein synthesizing machinery, or we can say it is these two molecules in association with the mRNA, it helps in bringing out the translation of the specific kind of protein. Okay, so this is what the what you call the transfer RNA does. Okay, so it first binds with the transfer RNA binds with a specific amino acid. Okay, what decides this specificity? It is the anticodon sequence which is present on the transfer RNA. We'll be looking into in when we study the structure. Okay, so the tRNA, it first binds with the specific amino acid, and this particular reaction, that is the binding of the amino acid with the tRNA, it utilizes energy. Okay, so it utilizes ATP. Then this kind of a what you call tRNA which is bound with to the specific amino acid, we call it as a what you call amino acetylization, okay, or amino acylation, or even we call it as a tRNA charge. Okay, it is charged tRNA we can call, right? This this tRNA bound to the amino acid, the charged tRNA, it moves to the ribosome, right, where the mRNA it is ready for getting translated. So it moves to the mRNA at the protein synthesizing site, right? And finally, the tRNA, the specific sequence on the tRNA, that is known as the anticodon, it binds with the specific sequence on the mRNA, which is known as the codon, right? So anticodon and the codon, as the name indicates, it is complementary. So it binds by the bonding, hydrogen bonding. Okay, and this actually brings about the base pairing, complementary base pairing, and this helps in the proper orientation of amino acids, and this is what happens repeatedly in the process of translation. Okay, so we saw that the tRNA binds with ribosome, binds with the amino acid, and this process require ATP, and the process which it is known as a charging of the tRNA or amino acylation of the tRNA. Okay, and this process, amino acylation of tRNA, it is catalyzed by the enzyme amino acyl tRNA synthetase. Right, this tRNA, the charged tRNA, it serves as a source of amino acids for the translation or for the synthesis of the peptide or the polypeptide chain. Right, now, we already know that there are 20 amino acids which are used for the synthesis of proteins, or for 20, there are plenty of amino acids, but among them only 20 amino acids comes into play for the synthesis of proteins. Right, and hence there are at least 20 different kinds of tRNA that can bind specifically to these 20 amino acids. Okay, so tRNA is they include the smallest of all the nucleic acids, right? And it contains maybe like 70 to 100, we can say 74 to 95, about 74 to 95 bases. They are the smallest among the three major RNA, and obviously DNA being the biggest, the largest. Right, now another important feature about the tRNA is the folding of the primary chain. Okay, so here you can see this is the primary chain over here. Just 1 minute. Okay, so here you can see the primary chain. This is a primary chain. But what you can see is there is a complementary base pairing within the chain. There is intra-chain base pairing can be found in the tRNA, okay, in the primary structure itself. So there is a folding of the primary chain, and the folded tRNA molecule, it appears, you can see with bends, with loops, with stems and all, right? So this bend and the loops and the stems, it usually result in the formation of a structure which is similar to the clover leaf. Okay, this is what is known as a clover leaf. Okay, so hence this structure is otherwise known as the clover leaf model of the tRNA. So the secondary folding, it maximizes the intramolecular hydrogen bonding and the base pairing between the adjacent loops. Here you can see one important feature. Here you can see that as already mentioned, this primary structure of all the tRNA molecules, they allow extensive folding and intra-strand complementary base pairing. Okay, and it results in the formation of a secondary structure, and it is this secondary structure which is similar to the clover leaf. Okay, so clover leaf, that is what is referred as a clover leaf model. All tRNA molecules contain 4 main arms. You can see here the 1, over here the 2, the 3, and the 4. Okay, 4 main arms are found in all the tRNAs, irrespective of what kind of amino acid it is going to bind with. And these are the acceptor arm, then we have the T-C loop, the T-C arm, the anticodon loop and anticodon arm, and the D loop or the D arm. Okay, the acceptor arm, it is the part which actually accepts the amino acid. That is how the name has come, the acceptor arm. The acceptor arm, it terminates in a very specific sequence of nucleotide. It is referred as a CCA, a specific nucleotide, nucleotide sequence. Okay, and these the nucleotides, the CCA in this sequence, it is added post-transcriptionally. That is, after the tRNA is produced from the tRNA gene on the DNA, it is a post-transcriptional modification. Okay, after the tRNA is produced, it is added post-transcriptionally, these 3 nucleotides. Okay, and it is added by a specific nucleotide transferase enzyme, right? So we have CCA being added at the 3' end of the tRNA chain. Fine, and here you can see it is this CCA, this is the sequence actually binding with the amino acid. Okay, so this tRNA, the specific amino acid is attached to the onto this 3' hydroxyl group of the adenyl nucleotide nucleotide of the acceptor arm. Okay, then we have as the D arm, which is referred as the what you call the dihydrouridine arm. Okay, the reason is it contains a very unique what you call nucleotide, pyrimidine nucleotide. It is the hydrouridine dinucleotide. Okay, so the presence of the dihydrouridine dinucleotide or dihydrouracil gives the name D loop. And this dihydrouridine arm, it contains a basal stem, you can see over here, and a loop over here. Okay, the loop is usually composed of 8 to 12, 8 to 14 bases, nucleotide bases, and it is this loop which contains the hydrouridine dinucleotide, or it is dihydrouracil. The DHU loop, it is essential for the binding of amino acyl tRNA synthetase enzyme. Okay, so the enzyme which is responsible for binding of the specific amino acid onto the acceptor arm, it is being recognized and it is being bound to at the like bound to with the help of the D loop, right? The enzyme which is concerned with the recognition and activation of the amino acid. Now then we have is the anticodon arm. Okay, the anticodon arm has two parts. It is the basal anticodon stem. This is the stem. Okay, and the anticodon loop we call, right? The loop, it is usually formed of 7 nucleotides. You can see 5 to 7 nucleotides, and among that you can find a 3 nucleotide sequence over here, which is referred as the anticodon. And it is this anticodon which is the complementary base sequence, complementary to the base sequence which is complementary to the codon present on the mRNA. Okay, you may have studied the genetic code in your lower classes, right? Genetic code or the codon. It is a triplet code. That is a codon. It is formed of 3 sequences, 3 consecutive sequences, right? So the anticodon, it is complementary to the codon on the mRNA. So during the translation process, this anticodon on the anticodon loop, it recognizes the corresponding complementary codon on the mRNA and binds it with the what you call the hydrogen bonding. So via hydrogen bonding, the sequence on the anticodon, it binds with the genetic code on the mRNA. So the anticodon loop, it is also referred as the mRNA recognition site. Okay, so it recognizes the codon sequence on the mRNA, and hence this loop is known as the mRNA recognition site. The codon-anticodon pairing, it helps in an orderly arrangement of the amino acids in the exact sequence as against the what you call the codon sequence on the mRNA, and hence result in the formation of the specific protein which is to be produced as a result of the transcription translation process of that particular gene up from the DNA. Okay, so it is very important. The identification of the codon on the mRNA with the help of the anticodon, it is very important. Okay, now the next one we have is the T-cyc loop. Okay, T-cyc, here you can see the sequence, right? The thymine, the pseudouracil, and the cytosine. These 3 what you call the nucleotides are found in the same sequence, 5 prime, T-cyc 3 prime. In that orientation it is found, and here the T-cyc arm also have a basal stem, you can see over here, and a T-cyc loop. The T-cyc loop, it is formed of nearly 7 bases, 7 nucleotide bases, with the presence of the pseudouridine, and that is how the name has come. Okay, and this particular loop, it is involved in binding of tRNA with the ribosome during translation. So D loop, it is involved in the what you call binding of the enzyme which is required for the binding of tRNA with the amino acid. Anticodon, it is involved in the binding with the mRNA, specifically at the codon region. And T-cyc loop, it is involved in binding of tRNA with the ribosome during translation. So it is known as a ribosome recognition site. Okay. Now, this is what the whole, what you call the clover leaf model, main features of the clover leaf model. So the common features of the secondary structure or the clover leaf model of all the cytoplasmic tRNA is as follows. There are 7 base pairs in the amino acid stem, and 3 or 4 base pairs in the dihydrouracil stem or the DHU stem, then 5 base pairs in the anticodon stem, and 5 base pairs in the TC stem. Okay, so these are the various features upon which the secondary structure of tRNA is being elucidated. Okay, here you can see the 3D structure as well as the 2D structure. The picture on the left side here, you can see this is a 3D structure. Okay, three-dimensional structure, and this is the clover leaf model, the 2D structure. Okay, so the flattened